Good afternoon, all my dear students. Welcome to the third semester of your course with the paper MSCH 302. The paper name is Spectroscopy. The spectroscopy is an analytical technique which is useful for the identification and analyzing the sample. The paper objective is to learn this analytical technique with the various terms involved and the various types of the spectroscopy which can be used to analyze different kind of the compounds that are synthesized in the lab or which found in nature. Spectroscopy word consists of the two words, that is spectrum plus scopy. The scopy meaning is to study and the spectrum meaning here, that is electromagnetic spectrum. If we talk about an ordinary spectrum, we can understand when a light is passed through a prism, it dispersed into various colors, and that is called the spectrum, which splitted into different colors. But here the spectrum definition is the broad definition, that is of electromagnetic spectrum. In this lecture, we are going to cover the introduction of the spectroscopy with the definition, what is the goal of the spectroscopy, then the spectrum, electromagnetic radiation, electromagnetic spectrum, various types of the spectroscopy, the instrument used to analyze the spectroscopy, that is the spectrophotometer, the various types of the spectrophotometer, and the advantages of the technique. What will be the learning outcome of this lecture? The students will able to understand about all the basic terms used in the spectroscopic technique, like the spectrum with the definition, various types, the instrument features, and the advantages. So let's start what is spectroscopy. As we understood that this consists of the two words spectrum plus scopy to study out the spectra. The spectroscopy is the study of the interaction of the electromagnetic radiation and the matter. The matter can be the atoms, molecules, or the ions. When the radiation falls to the matter, some of the radiation are absorbed by the atoms or the molecules. Some are transmitted, some are scattered. So on the basis of the response of the matter, when the radiation falls into it, on the basis of that response, we identify that sample. It is the first and foremost technique for identifying the sample. And then we can also analyze the amount. The quantitative analysis can also be done further. What is the goal of the spectroscopy? We should able to understand how the light interacts with the matter and how you can use this to quantitatively understand your sample. First, we identify the sample and then we quantify the sample. Understanding the spectroscopy is the same way as you understand other common tools of measurement, like watch to measure the time or ruler to measure the length of the object. It is as same as these measurement tools. For identifying the samples, you can use this spectroscopy. See that spectroscopy is a set of tools that you can put together in a different ways to understand the system, as well as you can solve it further. What is spectrum? The spectrum is a plot of the amount of the light absorbed by a sample versus the wavelength of the light. The amount of the light absorbed is called absorbance. This is the common spectrum, which is called the visible spectrum here. Here you can have the white light. When is passed through a prism, it's split into different colors. So at a particular wavelength, these colors we are able to see, that is Vibgyor. If you just pass this light to the matter, any of the wavelength of the light is not absorbed in this spectrum. Only the simple light is split into different colors of this particular wavelength. So you should know that visible light has these wavelength of the light in it. So this is only the visible spectrum when this light is passed through the sample. So some of the wavelength of the light in the visible range is absorbed by the sample, and you get the dark area under which that wavelength is absorbed. So we can easily understand the amount of the light which is absorbed by the sample. That is called the absorption spectra. Understood? And the visible range of the radiation is 400 to 800 nanometers. Okay. So this is the range of Vibgyor. 400 to 800 nanometers. So you can able to see this is the range 400, starting from here violet, indigo, blue, green, yellow, orange, red, and red up to this 800 nanometers. So we can easily understand this visible spectra, and one kind of the particular wavelength in this range is absorbed by the sample can give the absorption spectra. So this is to be visible range, and we talk for more range of the radiation. This is the simple spectrum. What does a spectrum measure? The interaction of the light with the sample can influence the sample or the light. The method involves the excitation and detection. Here you can have the basic idea. The light, which is an electromagnetic wave, is passed through the sample, and it goes for the excitation by which we characterize the light after the sample, which is after passing the sample we characterize the light, or we characterize the change in the sample. The two ways are there. If we characterize the light after passing through the sample, or we characterize the change in the sample. The change in the sample which occurs in the different branch, and we just in spectroscopy we characterize the light after the sample. Light is an electromagnetic radiation. So what is electromagnetic radiation? The electromagnetic radiation is a form of energy that has both wave and particle properties. EMR is having the dual nature, always possesses two types of the property, that is wave as well as the particle. Many of the properties of electromagnetic radiation are conveniently described by means of classical model which embodies such as parameters having wavelength, frequency, velocity and amplitude. So various models has explained the property of electromagnetic radiation as a wave. In contrast to the wave phenomena such as sound, electromagnetic radiation requires no supporting medium for its transmission and thus easily passes readily in a vacuum. So it do not require any supporting medium and it easily transmitted and more readily in a vacuum. So this is one of the important property of the electromagnetic radiation which is used in the spectroscopy. So ordinary light is also electromagnetic radiation, as we understood that white light when passed through the prism, it dispersed into the various different type of the wavelength which is known as the spectra. Understood? So light is also the EMR. So including the light, when we put all the radiation in the increasing order of the wavelength or decreasing order of the energy or decreasing order of the frequency, is called electromagnetic spectrum. And this is only used, all the radiation in this electromagnetic spectrum are used to study the spectroscopy, are interacted with the matter. So as I have said, increasing order of the wavelength or decreasing order of energy. We all know the Planck equation, E is equals to h nu, where E is the energy, nu is the frequency and h is the Planck constant. So energy has the direct relationship with the frequency. If we see the relationship with the wavelength, E is equals to, if we put the value of frequency, that hc, that is velocity of the light, upon lambda, that is the wavelength. So energy and the wavelength are inversely proportional to one another. That's why if we are arranging the radiations in the increasing order of the wavelength, so it is said the decreasing order of the energy. The energy goes on decreasing from right to left and wavelength goes on increasing from left to right. And the frequency order is similar like energy. So as you are able to see the increasing order of the wavelength here, you are able to see that is in meters. It can be converted into nanometers. The visible range of the radiation come in between this spectrum, that is Vibgyor, which is having at the bottom side, you are able to understand it goes with the lower wavelength, that is 400 to 420, then 420 to 440 and goes on increasing up to red. that is having the higher wavelength, it is about 780 nanometers. So this is the Webigo range. We arrange this in the order of increasing order of the wavelength. It can be understood by the decreasing order of the energy. And you can see after the visible, we have a longer wavelength is of IR, infrared region. If we are going from left to right, the visible, after the visible, we have IR, longer wavelength, IR, infrared radiation. Then we have a microwave radiation. You use microwave for cooking. So that microwave radiation are less energy wave we use, and having the longer wavelength up to 10 to the power minus 2 meters. Then we have more longer wavelength, the radio waves having the less energy, 10 to the power 2 to 10 to the power 4. So these are the longer wavelength or less energy radiations, IR, microwave, radio wave, used in the spectroscopy. Visible is also used in the spectroscopy. Now we come to the left side where the energy goes on increasing. After the visible, the high energy radiation, the lesser wavelength, that is UV, ultraviolet, which is used in the spectroscopy. Then more high energy you are able to see with the lesser wavelength, that is X-rays, and very high energy radiation, that is of gamma ray. So up to the ultraviolet light, we use the spectroscopy technique using UV radiation and interact with the matter. Up to UV, we use these radiations to interact with the matter and give out the identification. But the X-rays and gamma rays are not used in the spectroscopy. So after UV, high energy radiations are not used in the spectroscopy. If you use X-ray and just interact with the matter, your whole atoms and the molecule present in the matter just destroy it, because too much high energy it is. So the matter collapse. That's why we do not use high energy radiation in the spectroscopy. So this electromagnetic spectra we should understand while we are studying the spectroscopy. So on the basis of the radiation used from this electromagnetic spectra, we interact with the matter, and then what changes appear in the sample and the light after passing the sample to be analyzed, and that give the identification and the quantitative analysis. Now there are different types of the spectroscopy to study out, because when the radiation meets the matter, the radiation can be scattered, emitted or absorbed. After passing out the sample, when we analyze the light, so some of the light is absorbed, some are scattered, some are emitted. So on the basis of that, we have different types of the spectroscopy. The one of the important spectroscopy is the absorption spectroscopy, where whatever the light which is absorbed, that is to be analyzed. The second one is the scattering spectroscopy, and the third one is the emission spectroscopy. So in the absorption, absorbed light to be identified, and the scattered light which is studied out in the scattering spectroscopy, and emitted light to be studied out in the emission spectroscopy. Most of the spectroscopy, we characterize how the sample modify the light entering into it. In the absorption spectroscopy, let's see. When we pass the light to the sample, it goes on absorbing that light, and absorption takes place in the form of photons on the packet, that is E is equals to h nu, and goes from the ground state to the excited state. By the absorption, it reaches from ground to the excited state, and this causes the absorption, and we study the light. We measure the absorption of the light at a different frequency or the wavelength. At a different wavelength and a frequency, we pass that wavelength pass to the sample, and it goes to the excitation or not, it is to be analyzed. So we directly pass the radiation, particular frequency or the particular wavelength is passed to the sample. It is not all the wavelength or all the frequency of the radiation is passed simultaneously. No. One by one particular wavelength is passed and see its absorption. Like here in the graph you have this absorption spectra, that this particular wavelength on the x-axis we can measure this particular wavelength is highly absorbed, which is giving this peak, gives the absorption spectra, and somewhere the less absorption takes place of this wavelength and this wavelength. That can be analyzed in the absorption spectra. So in absorption spectra, by absorbing the light, electron goes to from the ground state to the excited state. And if we study out the emission spectra, the higher energy electron comes to the lesser energy, comes to the ground state by emitting the radiation, again in the form of photons. So here arrow is representing the radiation are absorbing and radiation are emitting. So from the higher energy state, when comes to the ground energy state, the atoms or molecules give up the energy in the form of packet, that is h nu, and which is called the emission. So excitation reduces the emission of the light from the sample, usually of a different frequency. So emission can be taken place from the each one of the excited states, and it can cause the emission in the form of fluorescence. The various excited states from where the electron comes to the ground state depend on that. So the fluorescence emission, phosphorescence emission, Raman scattering, which involve the vibrational transitions. So on the basis of the emission of the light, we can easily identify the sample. So there are the main two types of the spectroscopy which are used under study, that is absorption and the emission. Generally, the absorption spectroscopy is generally used because it is easier to give up the energy of the electron to the matter instead of putting it with the high energy state and see its emission. So it is easier to analyze the absorption spectra as compared to the emission spectra. After understanding the types of the spectroscopy, now we should learn what is spectrophotometer. The spectrophotometer, which is also called spectrometer, it's an instrument used to analyze the spectroscopy. The different type of the spectroscopy like absorption spectroscopy, emission spectroscopy, we can study by using the instrument which is called spectrophotometer. It is also called the spectrometer. The spectrophotometer consists of the main parts and the features, that is the light source, a monochromator, the sample holder with the reference holder and light detector with the amplifier and recorder. So one by one we should see each one of the parts in detail. The light source is the electromagnetic radiation source you should know to provide for the interaction of the matter. The function of the light source is to provide a sufficient light of the different wavelength. The light source typically yields the high output of polychromatic light over a wide range of the spectrum. So having the wide range of the wavelength of the different electromagnetic radiation it should consist of, that should be the light source. If we want to give the visible range of the radiation, so we should use that type of the source which involve all the radiation having in the range of visible Weber, it should be there if we are providing the visible range. If we are providing the UV range, so that source consists of all wavelength, that is to be said to be a polychromatic. Polychromatic means many wavelength of the radiation it consists of. So the source should contain all the wavelength of the radiation that should be passed to the matter to analyze it. So the main function of the light source is to provide the sufficient radiation that can be interact with the matter. Like in UV spectroscopy, you have to provide the UV light source that is of the hydrogen discharge lamp. That is more suitable light source of UV. For the visible, it is tungsten filament. So the particular light source to be selected according to the electromagnetic radiation which we are using to study for the interaction. Now this is one kind of the light source, that is hydrogen or deuterium lamp. For the ultraviolet region, hydrogen or deuterium lamps are frequently used. As I have said, the hydrogen discharge lamp as well as the deuterium lamps, their range is approximately 200 to 450 nanometers. Deuterium lamps are generally more stable and has the long life, about 500 hours. And this lamp generates continuous or discontinuous spectral. So here you can able to see this hydrogen or deuterium lamp which is suitable for giving the UV range of the radiation. Likewise, for the visible, we should select the tungsten filament. So the light source is very important in the spectrophotometer, which gives this wide range of the radiation, and it should be stable for giving out the radiation. So the next part of the spectrophotometer is the monochromator. The monochromator accepts the polychromatic light which is coming from the light source or a lamp and converts into the monochromatic light as an output. So only at a time the one type of the radiation of the particular wavelength is passed to the sample. Monochromator consists of the three parts: the entrance slit from which the polychromatic light come up, the exit slit, and the dispersion device which converts the polychromatic to the monochromatic. The examples of the monochromator can be the filters in the equipment which is used. So the filters or we can say the prism. Prism is also the example of the monochromator. Prism, filter, even gratings are used in the spectrophotometer nowadays for converting this polychromatic light into the monochromatic light. The next feature is the sample holder or the cuvette. The cuvette is a kind of a cell, usually a small square tube sealed at one end. It is made up of plastic, glass or the optical grade quartz. For the UV radiation, the cuvette is made up of quartz, and for the IR radiation, we use the glass cuvettes specifically. It is designed to hold the sample for the spectroscopic experiments. The cuvette should be as clear as possible without impurities that might affect the spectroscopic reading. So the cuvette which is used in the spectrophotometer, it should be clean when we put our sample for the analysis, so that it should be not affect the spectroscopic reading. So that material should be used to make the sample holder that not absorb that particular radiation. So this is, here you are able to see the diagram of the cuvette. These are made up of quartz when we use in UV, or they are made up of glass when we use in IR. So different kind of the material on the basis of the type of the spectroscopy or type of the radiation which we are using in the spectroscopy, it should be selected on that purpose. Why should we use quartz cuvette for measuring the absorbance in the UV range and plastic cuvettes or the glass cuvettes in the visible range? That the question always come up in the mind, as I have said, why there is a selection of the material to be made up of the sample holder. Either we are using different kind of the spectroscopy. The answer will be the same, that quartz do not absorb the ultraviolet radiations. So when we are studying the UV visible range of the radiation, UV and the visible range of the radiation are not be absorbed by the quartz. That's why we are using the cuvette made up of quartz in the UV visible spectroscopy. While in visible range of the radiation, it do not absorb by the plastic or the glass. That's why we have chosen the cuvette material or the sample holder material. The reference holder, it is also made up of the same material. If we are using for UV visible, it is also made up of the quartz, and for the IR it is made up of plastic or the glass. So reference holder also have the same material as the sample holder possesses. Now the next part is the detectors. The detectors convert the energy of the radiation into the signals. So the name of this particular component provide the function that it accomplish, it detects and measure the light intensity. So it convert the energy of the radiation into the signal. The light intensity is proportional to the current, and if this particular component is not working properly, the testing results will not be reliable. So detector is again an important part of the spectrophotometer, which is very much essential, because the light when passed through the sample and when it is coming out, the transmitted light, that to be detected by the detector. So it converts into the signals which comes in the form of the current, and that is amplified and recorded. in the recorder unit. So the amplifier and recorder are attached towards this detector. So whatever the signals which are producing by the detector, it can be recorded in the recording unit, and recording unit is generally the computer monitor. So computer in the monitor, it is goes for recording that signal which give us the identification of our sample. So the detection is an important part of the spectrophotometer. So this is a diagram of all the parts. They are connected to one another. This is the source, which is coming to the monochromator. Here you can able to see this is the monochromator, which can be here the prism, and the light is coming through this entrance slit, which is having this monochromator dispersion device, and then the light from the monochromatic radiation, it converts all the polychromatic radiation to the monochromatic into this sample here, and that detection is done by the detector. So detector plays the role of conversion of all the radiation into the energy, which is finally recorded to the recording unit. And this how all the parts are attached to it. The source, then we have a monochromator is having the three devices: entrance slit, dispersion device, and the exit slit. Then we have the sample attached. Somewhere we have the reference. At the same time we have the reference holder also with your sample, which gives more accurate information. So we will read out the reference cell into the spectrophotometer in the types of the spectrophotometer further. And finally the detector goes on detecting, and it is recorded in the recording unit. So this whole is the equipment, how it works, and we can easily analyze the spectroscopy. Now we see the various advantages of this spectroscopy. It is a simple method of the analysis like the other measurement tools, because only we have to put our sample in the equipment and just pass to the source of the light, and we can easily goes for identification of the compound. The most important thing of this technique advantage, very less amount of the sample is used, because before the invention of the spectroscopy technique, we used to analyze the sample on the basis of their properties and the chemical reactions involved in it. So there is a big problem arises when we synthesize our sample and the whole sample just consume in identification on the basis of the reaction and the properties we determine. After the invention of this technique, very less amount of the sample, very less, it is 0.1 milligrams, yeah, very a small crystal part of the sample is used to analyze the sample, and it gives very accurate and reliable results. Before we are getting the results that should not be so accurate, it can be varied with the procedure, because so many error comes out when we are doing on the basis of their properties. So that becomes very difficult to have the accurate results. But the spectroscopy give, with the very less amount, we have very reliable and the accurate results. It is cheaper in the long run. Once you have your equipment or the instrument, and you can go for analyzing many of the samples. So it's a, you can say it is cheaper in the long run once you invest the money and you can take the advantage of the analysis of the sample. So very advantageous technique it is, and easy to analyze. So this is all about the basic idea and the introduction to the spectroscopy technique, including the various terms, types, and the instrument. In the next lecture, we should go for further detailing of the types of the spectroscopy, including the types of the spectrophotometer also. Till then, thank you and take care.